Okay, hello to all of you. Let's discuss chapter number 15. And chapter number 15 is about it's about the sex-linked inheritance. So let me go into slideshow more. Okay. So basically in chapter number 14, we talked about Mendelian and non-Mendelian inheritance. We talked about Mendel's crosses. We talked about monohybrid cross, dihybrid cross, and basically we talked about the term which was being used by Mendel was heritable factors, because at that time there was not much knowledge about genes, there was not much knowledge about cell divisions, and so people didn't knew exactly what exactly is going on at the molecular level. So basically Mendel used the term heritable factors, and after that, when the more developments came, people did more research, and they came to know that these heritable factors, which Mendel was using the term, and he said that these heritable factors are actually passed from parents to the offsprings. So they are actually, now we call those heritable factors as genes. And we also came to know that these genes, which are made of DNA wrapped over proteins, they are located on the chromosomes. So now we know that these genes, which he was calling them as heritable factors, they are basically located on chromosomes. And now we can actually, like these are the chromosomes, and we can we can tag these genes with some fluorescent markers, and we can show all these things now. And in around 1875, the cytologists, they basically worked on the process of mitosis, and then in 1890s people came to know about the cell division, meiosis. And then people started coming to know that these heritable factors, which we now know they are genes, they are located on the chromosomes. And around 1902, these are the two scientists, Sutton and Boveri, they independently noted, and they developed the chromosome theory of inheritance. And now we know that these genes, they are located on the chromosomes. So I have already shown you this cross in terms of alleles in my last lecture, and I have shown you how these alleles, which are the two variants of the gene, they are located on the chromosomes, and how these alleles, they get separated during the process of meiosis, and that results in the formation of gametes, and there lies the law of segregation of the Mendel. So we have discussed all these things in the last chapter, and we also discussed about the law of independent assortment. So how one chromosome pair is aligned, it is all independent of the other chromosome pair. So we have discussed all these things in the last chapter. So this is basically showing you again the same thing, that this is Mendel's dihybrid cross. So in the PowerPoint, the dihybrid cross is not shown in terms of alleles and how these alleles, they get separated in the during the meiosis, and it results in the formation of gametes. This slide in the PowerPoint exactly shows what I have shown on the whiteboard in the last chapter. So basically you can see that these are the chromosomes, and here are the alleles. So this is the dihybrid cross, yellow and the round seeds, green and the wrinkled seeds, and you can see that this is one chromosome, this second chromosome, and it has alleles for yellow color. This chromosome has an allele for yellow color. This chromosome also has an allele for yellow color, and this chromosome, they have alleles for the round texture. So we also showed that these genes, they're present on different chromosomes. And this is the seeds, which are green and wrinkled. And so you can see that this is the chromosome with the allele for the green color. This is another chromosome for having allele for the green color. And these are the chromosomes with the alleles for the wrinkled texture. And they undergo meiosis, and we talked about by the process of meiosis, gametes are formed. So this cell will be forming gametes which will be having capital Y, capital R, we discussed last week, and from this the gametes will be small y and small r, and when they fertilize they are going to form the offspring in the F1 generation, which is going to have capital Y, capital R and small r. And then these individuals which are having capital Y, capital R, small r, when they fertilize, when they cross, then the next generation is formed, that is F2. So first of all, what kind of gametes will be formed from this individual? So basically the alleles are different here. So one allele is capital R, another is small r, then capital Y and small y. If the chromosomes are aligned in this way, then the gametes formed will be these. But as we discussed, the alignment of chromosomes can be other way around also, which means the blue chromosome can be on this side, red can be on this side, and then these are the kind of gametes which are possible. And then in F2, these all gametes they fuse, and then they form these kind of offsprings in this ratio, 9:3:3:1. So this is all discussed in last week's lecture. So this is all on the basis of that these genes, they are located on the they have a specific location on the chromosome. So basically then came another scientist called Thomas Hunt Morgan. He was the one actually who did experiments with fruit flies, and he actually his experiments actually gave evidence that the chromosomes are the location of Mendel's heritable factors. So Morgan chose Drosophila melanogaster. This is a fruit fly, and he was also lucky, like Mendel, that he also chose a great model. Like Mendel's model was peas. So there were many advantages of using peas. So similarly, there are many advantages of using the Drosophila, the fruit fly, because they produce many offsprings. A generation can be bred every 2 weeks, and they have only 4 pair of chromosomes. So basically, Morgan basically was working on these fruit flies, and what he observed that in a group of fruit flies which normally have their eye color is red. So that is so normally these fruit flies, their eye color is red. So we call this character as a wild type character. So in the group of the normal flies, he found one mutant fly, and that mutant fly had a white eyes and it was male. So that was the finding of the Morgan. So what he did with that? So this is wild type fly. The color of eyes is red, and this is the mutant fly. The color of eyes is white. So basically what he did, and this experiment proved that these heritable factors of Mendel, which we now call genes, they have a specific location on a chromosomes. So what he did actually, so in one experiment, Morgan mated male flies with white eyes, which means they're mutant flies, with female flies with red eyes. What he found, in F1 generation, all flies had red color. The eyes were red. So what he did, he took these flies and he mated them. And then in the F2 generation, he got red to white eye ratio in the flies was 3:1, which means 3 flies had eyes red in color, and 1 fly had eye color white, and only males they had white eyes. Now let's see what does it all mean. So on the basis of this experiment, basically Morgan on the basis of these results, he reasoned that the white-eyed mutant allele, it was located on the X chromosome. So this finding supported the chromosome theory of inheritance, which means the genes are located on specific chromosomes. Okay, so now this is the cross of Morgan, which he did, and now let's see how he showed that. So on the basis of this experiment, he reasoned that the mutant allele is located on the X chromosome, and that proved the chromosome theory of inheritance. So now let's see. So what he did in the P1 generation, the parental generation, he took the female fly who had eyes of red color. He took a male fly, eyes were of white color. He crossed them. Okay. In the F1 generation, he got all flies red in color, and he took these flies and he mated them. In the next generation, he got the flies, and these flies, they had red to white color of the eyes in the ratio of 3:1. So now let's see. So basically, he took the female fly with red eyes. So which means that this female, if she's having red color, so it means that both the alleles on the X chromosome, it's a female fly. So I hope you know that there is a pair of sex chromosome in the individuals, and XX is for female and XY is for the males. So these are the two sex chromosomes in the female fly. Both chromosomes are X. Both the sex chromosomes are X. So the allele which is present on the X chromosome is a normal allele, and it is designated as W+. And then on the another X chromosome, the allele is normal allele, it's also W+. And this is a male fly with white color, so it means this fly has a mutant allele, and the mutant alleles are written like this. That's written as W. So normal alleles are written as W+, mutant alleles are written as W. So this is a male fly, so the sex chromosomes are X and Y. So this is the Y chromosome, and this is the X chromosome. So the mutant allele is present on the X chromosome. That was his reasoning after he got these results. And now we are actually looking in terms of this reasoning. So this is the female. So in the meiosis, this female is going to make gametes with normal allele. So after meiosis, female is going to make eggs, and all eggs will have X chromosome, and they will have normal allele present. This is a male fly, and the sperms which will be formed by the male will be of two kinds. One will have Y chromosome, another will have X chromosome, and on the X chromosome there is a mutant allele. So when these eggs, they fuse with the sperms, these are the individuals possible. So when this goes with this is X chromosome, this is X chromosome. X and X, it's going to be a female. One chromosome is having normal allele, another is having mutant allele. So this female is having one mutant allele, one normal allele, which means that this female is going to be a carrier. So when there is a heterozygous situation, one allele is normal, another is mutant, then that individual is a carrier. Now this is X chromosome, this is Y. So X and Y. X and Y means the fly is going to be male. So this is the normal allele. And on the Y chromosome, no allele, because usually Y chromosomes are very small chromosomes. They don't carry much genes. So this individual is going to be a male, and because on the X chromosome it has a normal allele, this male is also going to have red color of the eyes. So this is female, this is male, both have red color. So it means that in the F1 generation, all the individuals, they will have red color of the eyes. So he took these individuals and he crossed them, and that formed the F2 generation. So this is a female, this is a male. So this female is going to form these two kinds of eggs, X chromosome, X chromosome. But this X chromosome has normal allele, this has a mutant allele. This is a male. So this is going to form two kinds of sperms, X chromosome with the normal allele, and this is Y chromosome. So when this crosses with this, it's going to be X chromosome with X, it's going to be a female. Both alleles are normal, so this female will be having red eyes. Now when this goes with this, X and Y, this is going to be a male, this is going to be son. X chromosome has normal allele. This is Y. So this is again going to be a male, this is going to be a son with red eyes. Now if this goes with this, it's going to be a X chromosome with X chromosome, female. One is a normal allele, other is a mutant allele. This is normal, this is mutant. So this is again going to be a female with red eyes, but she is a carrier, she is heterozygous. And when this goes with this, so this is X chromosome, when it goes with this Y chromosome, it's going to be a male, XY. This is mutant allele, this is Y. So this is going to be a male with white eyes because this allele is mutant allele. So on the basis of this cross, so on the basis of his results, he reasoned, and now we have followed his reasoning. So that actually shows us that the mutant allele is actually located on the X chromosome. So this discovery, so Morgan's discovery of a trait that correlated with the sex of flies was key to the development of chromosome theory of inheritance. Okay, so now let's talk about the sex chromosome in humans. So basically in humans, if it's a female, then the sex chromosomes are X and X. But if it is a male, one chromosome is X and other is Y. So you can see the X chromosome is a very big chromosome, Y chromosome is a very small chromosome. So and X chromosome is a big chromosome, it carries a lot of genes and they code for many proteins. But the Y chromosome is basically a small chromosome, and Y chromosome codes for very less genes, and Y chromosome is basically for the sex determination. Okay, so Y chromosome makes male a male. The short segments at the ends of Y chromosome are homologous with the X chromosome. So the short segments at the end of Y chromosome are homologous to some segments here on the X chromosome. That's why they pair up. They form a homologous pair. A gene on the Y chromosome called SRY, sex determining region on the Y chromosome, is responsible for the determination of the testes in the embryo. So this gene is responsible for the sex determination in the males. So the gene that is located on either sex chromosome is called a sex-linked gene, and the genes on the Y chromosome, they are called Y-linked genes, and the genes on the X chromosome, they are called X-linked genes. So Y chromosome, as I said, is a very small chromosome. It has only 78 genes and they code for 25 proteins, I'm sorry. They code for 78 genes and they code for 25 proteins. And the human X chromosome contain around 1100 genes. So X chromosome is a big chromosome, Y chromosome is a small chromosome, and it is mainly responsible for sex determination. So X-linked genes, they follow a specific pattern of inheritance. So basically, like in the females, there are two X chromosomes. Suppose a mutation takes place in one of the allele on the X chromosome. The effect of that mutation is not going to show up, because in females there is another X chromosome, because the other X chromosome will be having a normal allele on the other X chromosome. So but in the males, because in the males there is one chromosome is X, another is Y. So if there is a mutation which takes place, on the X chromosome and the allele get mutated, and if the allele get mutated on the X chromosome, the second chromosome is a Y chromosome. In the males, they do not have two copies of X chromosome. They have only one X chromosome, other chromosome is Y. So if some mutation takes place on the X chromosome, it does not have the second copy, because it does not have the second X chromosome. So if mutation takes place on the X chromosome in the males, that mutation is going to show its effect. But in the females, it's not going to show its effect unless and until on both the X chromosomes that allele gets mutated. So for a recessive X-linked trait to be expressed, a female needs two copies of the allele, as I said. So in the females, if mutation takes place, they have another copy of the X chromosome which has a normal allele. So in that situation, female is a carrier. She is not going to express the gene. It's not going to show the effect of the mutation of the allele, but she will show the effect only when both the alleles they get mutated. A male, as I said, it needs only one copy of the mutated allele to show its effect, because males, they have only one X chromosome. So X-linked disorders are much more common in males than females. Okay, so now we can actually discuss some of the crosses, and it's important to understand these kind of X-linked crosses, because you can get these kind of questions on your quizzes. So basically, we can see this is a female, okay, and capital N means normal allele. Okay, this is a female. Female sex chromosomes are XX. Both the alleles are normal. This is a male, XY, and the allele is mutated. So which means that this male will be affected male, because the mutant allele is on the X chromosome and the males do not have the second copy of the X chromosome. So if you see all dark area like this, it means this individual is suffering from the disease or is affected by the disease. If you see white, means normal. If you see like light shaded, it means this individual is a carrier. So if the mating takes place between this female and this male, let's see what will be the next generation. So this female is going to form the gametes, which are going to be eggs. They will have normal allele. All the eggs formed from this individual, from this mother, will be X chromosome with the normal allele. Now, from this father, there are two kinds of gametes will be formed, two kinds of sperms will be formed. One will be X chromosome with mutant allele, and then the Y chromosome. Another sperm with the Y chromosome. So if it processes with this is going to be a daughter. One allele will be normal, another will be mutated. So this daughter is going to be the carrier. She's not going to be affected, but she is the carrier. If this goes with this is going to be a son, and the allele on X chromosome is normal, so this is going to be a normal son. Now if this goes with this is going to be a daughter. One allele is normal, another allele is mutant. So this daughter is going to be again carrier. If this goes with this, it's going to be son, and this is normal allele, so this is going to be normal. So you can be asked, like, if this is the mother, this is the father, what kind of children they are going to form. So these kind of questions you can be asked. So in this case, if this is the mother, this is the father, the mother is the carrier, the father is normal. So these are the gametes which will be formed, and these are the gametes will be formed from the mother, these are gametes formed from father, and when they cross, this is going to be the next progeny. So which means that 50% are the daughters, 50% are the sons, and out of these, one daughter is normal, one is carrier. One son is normal, another is diseased son. So this is the same thing, same cross. It's a similar kind of a cross, which is between a carrier mother and the affected or the diseased father. So these are the gametes formed from the mother, and these are the gametes formed from the father, and this is when they cross. These are the individuals going to be formed. The children will be formed. So these are the daughters. One daughter is carrier, another is affected daughter. This is a normal son, and this is the diseased son. So some disorders caused by recessive alleles on the X chromosomes in humans include diseases like color blindness, then muscular dystrophy, and hemophilia. Okay, the last topic in this chapter, we are going to talk about what kind of situations can arise if there is alteration in the chromosome number, or if there is some defect happens to the structure of the chromosome. So basically we know that as humans we have 23 pairs of chromosomes, or 46 chromosomes in us. So so the gametes are formed by the process of meiosis. So if something goes wrong and it results in the abnormal number of chromosomes, what effects can be there? So there is a process called non-disjunction which can happen during meiosis, and that can result in the formation of abnormal number of chromosomes. So let's see how the non-disjunction can take place. So you can see here, this is so this is, meiosis has two stages, meiosis 1 and meiosis 2. So if the non-disjunction, which is the unequal distribution of chromosome, if it takes place during meiosis 1. So in meiosis 1, what is happening? The homologous pair is separating. The homologous pair is separating. So the chromosomes, they separate. So suppose suppose non-disjunction takes place during meiosis 1, what happens? So it means that separation of chromosome will not take place here, and as a result of that, what will happen? One cell, so in after meiosis 1, two cells are going to be formed. One cell will have two chromosomes, and another will have 3 chromosomes. So these chromosomes, they are not separating here, and as a result of that, this cell will have 3 duplicated chromosomes, and this cell will have only one. It will have only 1 chromosome. So this cell will have 3, this will have 1. So what we are talking about, the process of non-disjunction. Non-disjunction means the unequal distribution of the chromosome. So if the non-disjunction takes place during meiosis 1, it will not let the chromosomes to separate equally, and as a result of that, what will happen? One cell will have, and remember that in meiosis 1 the chromosomes are duplicated. That's what these duplicated chromosomes are. One cell will have 3 duplicated chromosomes. Here they are just showing the separation of cystochromatids. One cell will have 3 chromosomes, the second one will have only 1 chromosome, and these are all duplicated, and they are now showing for the next meiosis 1, meiosis 2, the separation of cystochromatid. So this is the cell with 3 chromosomes, this is cell with 1 chromosome, and in the next phase, these cells will undergo meiosis 2. These cells will undergo meiosis 2, and that will result in the separation of the cystochromatids. And as a result of that, from this cell, 2 cells will be formed, and they will be having 3 chromosomes in them instead of 2 chromosomes. And this cell when it will form two cells, when this cell will form two cells, it will have one chromosome each. So these two cells will have one extra chromosome and these two cells will have one less chromosome. Now suppose non-disjunction is not taking place in meiosis 1 but it's rather taking place in meiosis 2. And as a result of that, after meiosis 1, the two cells which will be formed, they will have two chromosomes and two chromosomes will be duplicated. Now if suppose non-disjunction takes place at this point, then what is going to happen? So from this cell, the two cells which will be formed, they will be having two chromosomes, which is normal. But from this cell, when this cell will undergo meiosis 2, it will result in the formation of one cell with three chromosomes and one cell with one chromosome. So in this kind of a situation, two gametes or two cells which are formed, they will be having correct number of chromosomes, but one cell will have one extra chromosome and one cell will have one less chromosome. Okay, what is aneuploidy situation? Aneuploidy is a situation when the cells, they don't have the correct number of chromosomes, they have abnormal number of chromosomes. So aneuploidy results from fertilization of gametes in which non-disjunction occurred. So offspring with this condition, they have an abnormal number of chromosomes, or they have an abnormal number of particular chromosomes. Monosomic zygote means has only one copy of a particular chromosome, and trisomic zygote has three copies of a particular chromosome. A typical example of a trisomic zygote is in Down syndrome, because in Down syndrome, these individuals, they have three copies of chromosome number 21. Polyploidy is a situation in which, so there's a difference between aneuploidy and polyploidy. In polyploidy, basically the cells, they have complete extra sets of the chromosomes. It can be 3, it can be 4. So triploidy, when they have 3 sets of chromosomes, which means every chromosome. So aneuploidy is situation like chromosome number 21 has 3 copies, or chromosome number 5 has 3 copies, something like that. That is aneuploidy. Polyploidy is a situation when all the chromosomes, they have 3 sets or 4 sets. So triploidy is a 3 sets of chromosomes. Tetraploidy is a 4 sets of chromosomes. So it's a condition in which organisms, they have more than 2 complete sets of chromosomes, like the diploid situation. Like in humans, we have 2 sets of every chromosome. But if we have, by chance some situation happens, if somebody has 3 sets of every chromosome, that's a triploidy situation. Or if it has 4 sets of every chromosome, it is a tetraploidy situation. Okay, next we are going to talk about if some alterations takes place to the chromosome structure. Now it can be, the changes in the chromosome structure can be in the form of deletion, duplication, inversion or translocation. So deletion means, like you can see, like this is the chromosome and these are the genes on the chromosome. Suppose deletion takes place and this part gets deleted. Now the next chromosome will be formed, will be have a deleted, it doesn't have this gene here. So this is deletion. Duplication, suppose some genes get duplicated. Now suppose if BC gets duplicated, then on the chromosome we have BC and BC. So this is duplication. Inversion means, like this is the order of the genes. Inversion means the genes get inverted. Like here it is A, B, C, D, E, F, G, H. Now it has become A, D, C, B. So which means this part goes this side. So if this goes this side and this part goes that side. So this is inversion. Translocation is the exchange of a segment of chromosome from one chromosome to another chromosome. So basically it moves the segments from one chromosome to the another non-homologous chromosome. Now you can see here, this is one chromosome. So this is the sequence of the genes on this chromosome. This is sequence of genes on this chromosome. So these are not homologous chromosomes, they are non-homologous chromosomes. So if suppose this part of the gene, these genes or this part of the chromosome, it goes and become part of this chromosome, and this part of the chromosome becomes part of this chromosome. This is called translocation. And there are many diseases which are because of translocation of the chromosomes. So Down syndrome is an aneuploid situation in which there are 3 copies of chromosome number 21. So this is a child showing Down syndrome, and the reason is this chromosome number 21. You can see there are 3 copies. Then there are another syndromes like Klinefelter syndrome. It's a result of extra chromosome in a male. So usually the male chromosomes are XY. They have one extra X chromosome. Then there are some females which are called XXX chromosomes, sex chromosomes. So they have one extra XX chromosome. So they're healthy with no unusual physical features, although they are at risk of learning disabilities. Then monosomy X, which means the situation. So here is the situation when the females have one extra X chromosome. These are also called super females. Now this is a situation called monosomy X, also called Turner syndrome. They have only one X sex chromosome. They don't have two X sex chromosomes. They have only one. And these females, they are usually sterile. Okay, some of the diseases which are because of the disorders which are because of the structure changes, structure alterations which takes place to the chromosome. The first one is cry-do-chat syndrome, which is cry of the cats, and this is because of the specific deletion of in chromosome number 5. So some specific part of chromosome number 5 is deleted, and as a result of that, these individuals, they have this syndrome called cry-do-chat syndrome. So a child born with this syndrome is severely intellectually disabled and has a cat-like cry. And these individuals, they die in infancy or early childhood. Now the next disease we are going to talk about is chronic myelogenous leukemia, which is because of translocation of the genes between the chromosomes. So this is chromosome number 9, this is chromosome number 22, and some part of chromosome number 9 becomes part of chromosome number 22, and some part of chromosome number 22 becomes part of chromosome number 9. And that results that this translocation is responsible for the disease leukemia. So that's all in this